Department of Zoology, right? Yeah. Okay. And then he moved to Fribourg, and now he came back to the University of Geneva last September 2022 as a medical assistant. And today he will be talking about the consequences of interspecific hybridization. Thanks for accepting.
So, good afternoon. So today I'm going to present part of my research, and my main field topic is eco-evolutionary models. So during the first part of these presentations, I will say something about that, but also I will highlight why hybridization is considered a cryptic phenomenon. And I will show three computational approaches I have been working for studying hybridization. And then I will finish with the implications in conservation genetics and conclusions. So eco-evolutionary models provide great insights on the development of biodiversity patterns. A main feature of models is to recreate evolution multiple times. And this is very important because when you analyze a field observation, this observation is a result of a single evolutionary trajectory. But by developing a model, you could recreate this many times. So you could develop a model. So for instance, here you have a given phenotype in time t-1 and the same quantitative phenotype in. Okay. Voilà. Okay. Okay. Stop sharing. Okay. This one? No, it's not this one. Okay. Bad panic. Stop. Voilà. It's working. Okay. Is that okay? Good. So, as I say, by developing a models, so for instance, here, a quantitative phenotype in time t-1 and time t, you can start to make prediction about what may happen in these systems if, for instance, climate change the conditions for the near future. And there are powerful tools that have been developed during the last 20 years, but they are still limited in capturing the complex and nuanced conditions observed in nature, even more when regarding the new challenge imposed by the accumulation of genomes over the last decades. So I'm working in this domain, mostly in the hybridization phenomenon, which is known today as an important evolutionary force with an important role in the evolution of many plants and animal taxa. But there is a concern when hybridization is motivated by human factors. So for instance, domesticated organisms, because when they introduce these artificially selected genes during the domestication process to nature, they could change the behavior and the fitness of the wild organisms. So, and hybridization is considered a cryptic phenomenon with more cryptic effect than other ecological effects like predation or competitions, because even if the population size is the same, the individuals are not the same, genetically are not the same. And the consequence of this is different in each case study. And we know that because the introduction of invasive species, habitat modification, and climate change, we expect hybridization to be more important in the near future. And there is a poor knowledge regarding the biodiversity loss, the extinction of species, or alternatively, the new biodiversity, the new species influenced by this phenomenon. There is evidence of hybridization for all species for which we have one genome. So hybridization is known to be important today. A few years ago, during the last century, hybridization was not considered to be important in nature. It was considered to be something infrequent related to human forced interbreeding events. But today we know that hybridization has been important in the evolution is more likely of all species, because we have evidence of this for all species in which we have more than one genome. So today is an exciting time for studying hybridization. We can finally use model-based estimate in non-model organisms with accumulation of ecological and genomic data in public repositories. So I'm working in 3 computational approaches. The first one is my general model of hybridization. Here we have a quantitative phenotypes from blue to red, and this parameter gamma is the interbreeding rate between 2 population species. And so you could see over time how this phenotypic landscape change. The second approach is the splash software. So here we are simulating in a map the introduction of an invasive species, the colonization over time, and the interbreeding with the local organisms. The third approach is my R package GLADS. So here we are simulated the genomic information of individuals with the ecological information of those individuals over time. So because I don't have the full day for presenting my work, so I will present the 2 last approaches. And I will start with GLADS. So GLADS is an individual-based simulation framework. So here we have genotypes affecting phenotypes. These phenotypes affect the demographic rates, and these demographic rates affect the genotypes. And all of this is made in a way that we have interaction between the genomics of these individuals with the environment. So the idea is we have individuals represented by pairs of chromosomes, 1 pair or various pairs of chromosomes, and each color represents a different color pattern. So here we have a genotype that influences a phenotype, and this phenotype influences the fitness. So for each case study, we have to build a fitness function. And so in this case, we have the genotype interacting with the environment, resulting in a given phenotype, and this phenotype is influencing the fitness. So as an example, in this figure, on the y-axis, we have the fitness of the number of offspring, the average number of offspring for a population. The x-axis is a quantitative phenotype, and the two functions represent 2 different fitness functions for 2 different populations with 2 different phenotypic optimum. So this first phenotypic optimum and the second represents the phenotypic value that maximize fitness for a given population. So if both fitness functions are equal, they are considered to be under concordant selection, which is a kind of parallel selection. But if these 2 fitness functions are different, like in this case, they are considered to be under divergence selection. So here is an example, theoretical example, in which you will see that by little, the 2 different populations, red and black, reach the phenotypic optimum at the end of the simulations. So we will apply this to the metaphor of genomic islands of divergency or speciation with hybridization. So during the last decades, many genomes start to accumulate. So then the field of population genomics start to grow up, and many examples of genomic comparisons of the divergence between different populations start to accumulate. And this is an example in flycatchers. On the y-axis, we have the genomic differentiation. On the x-axis, we have the differentiation in the genome for different chromosomes. And you could see that the differentiation is not homogeneously distributed in the genome. There are some regions with higher level of divergence. So these are considered to be the islands of genomic divergence. So these islands were explaining at the beginning as an speciation with ongoing gene flow with hybridization. So the idea is these islands are genomic regions under divergent selection, and the rest of the genome is homogenized by gene flow. So we test this hypothesis by performing simulations. So first, concordant selections, and you could see that little by little, FST is a measure of genomic divergence, and this is a theoretical genomes on the x-axis. And after 100 generations, we have genomic islands randomly distributed. But if we simulate divergent selection, we expect higher genomic islands over loci influencing phenotypes, and particularly when these loci are physically linked, as in this case. But by performing these simulations many times with the same parameter values, we realize that sometimes we observe a pattern of genomic islands, but other times we didn't observe any pattern of genomic islands. And this was the same for concordant and for divergent selection. So I'm not showing that this result here, but I will tell you that the explanation of this is because the genetic identity of the source population before the split, before the divergence process is important. So depending on this genetic diversity, we expect to have this pattern or not at the end of the simulations. So over time, we expect this genomic island pattern to disappear. But all simulations have been showing now is simulations of two divergent populations in allopatry, without gene flow, without hybridization. And so we observe these patterns without a hybridization process, but if we include gene flow, we expect to observe the same pattern for longer. So, okay. And then we say, okay, what happened? What is the main effect of migration rate or gene flow? And linkage, so recombination rate. So higher genomic islands, which are the yellow color or the orange color, are expected under extreme linkage and low level of gene flow. So as a summary, to have a pattern of genomic islands, we need divergent selection and linkage or just drift. But at the very end, to have these patterns, the initial genetic diversity is important. Time has a negative effect because with time we expect everything as divergence start to accumulate, so the pattern disappear, and gene flow has a positive and negative effect because a small amount of gene flow allow to observe this pattern for longer, but too much gene flow delete erase any pattern. So then we apply all this framework in the case study of the silverized species complex on Southwest Pacific. So these islands are islands close to Australia. So we have 9 different islands, 400 individuals, and 3 million SNPs over the genomes. And with some islands with high level of gene flow or low level of gene flow. So here I just going to focus, I will focus on Aran Islands and mainland Australia. So this is the result of the genetic differentiation FST on the y-axis. And you will see that some genomic regions have higher level of differentiation. So then we simulate different recombination rates expected at the different chromosomes. And we estimate here the number of migration events from Australia to Aran Islands to explain the observed level of differentiation. So the dot line is the prior distribution and the solid line are posterior distribution in an averagation approach. And so we observe that with 1 or 2 individuals migrating each 100 years or 200 years or 100 years, we expect to have this pattern. So with no much gene flow, we can explain the level of differentiation between both populations. So finally, by doing many simulations, we observe that the distribution of FST or the distribution of differentiation across genomes is expected to be more close to the 0 values with that thicker distribution at high gene flow and early stage of divergence. And we expect to have many different levels of gene flow with late stage of divergence. So now I would like to explain the second approach, Splash. So Splash simulate in a grid of beams in a map the introduction of an invasive species and the colorization over time from time t to t+1 and the interbreeding with the local organisms. So it's a program designed to translate ecological information into molecular diversity. We have 2 different types of gene flow. So the vertical arrows represent hybridization, so the gene flow from one population or a species to another. And the second one, the horizontal arrows represent the migration or the gene flow within a given taxon, species or population. So why spatial dynamics is important? It's important because we have invasive species arriving with few individuals, and they are in range expansion, while the locals are at demographic equilibrium, and they have already colonized the full area. So because this demographic imbalance, we expect an asymmetric introgression, so the gene flow from one population to another to be much higher in the invasive gene pool compared to the local gene pool. And so we first observed this by simulations, but now it's confirmed with many hundreds of empirical studies. So it's considered today as a neutral expectation after any biological invasions with hybridization. A few years ago, I demonstrated that we also expect an asymmetric introgression within a given taxon, because we expect the introgression to be higher with the distance from the source of the expansion. So more far away you are from the source of the invasive expansions, more introgression you will have. And the opposite is expected for the local organisms. So I will explain all of this with two different case studies. So the first case study is about the asymmetry within a given taxon, so with the distance from the source of the expansion. And we will apply this to the Neanderthal case study and hybridization with modern humans. So a decade ago, when we have the first draft genome of Neanderthals, the researchers observed that about 2 to 3% of modern humans today have a level, modern humans today have a, around 2 to 3% of Neanderthal ancestry today. And if I take a random sample in this room, so for instance, me, even people even me that I have probably a Native American origin, I may have this Neanderthal ancestry, even if Neanderthal never exists in the American continent. But this is because the history of colonization of modern humans in Africa, in Europe, then Asia, and then the rest of the world, carrying these Neanderthal genes. So researchers also observe that the level of introgression is higher in East Asia compared to Western Europe, around to 12 to 20% higher. So most papers say, or most authors assume that this is because selection. So selection influenced this higher introgression in East Asia compared to Europe. But we say, okay, we may explain this with branch expansion. So we test this hypothesis. So we have this map, and the red colors represent the Neanderthal distribution. The sky blue represents the expansion of modern humans, and the yellow dots represent a few samples in France and samples in China. So we perform thousands of simulations. So we have a prior distribution of the interbreeding success rate parameter. So how frequently Neanderthals and modern humans interbreed. So this dotted line is the prior distribution, and the solid line is the posterior distribution in a Bayesian computational approach. And we obtain that a level of 2% of Neanderthal of interbreeding explain this level of introgression. So this is compatible with only a few admixture events. So I don't remember very well the numbers, but it's about like a very few, well, less than 100 admixture events during the full cohabitation times of Neanderthals and modern humans. So yeah. So in this figure, we have intermediate success rate. So we have intermediate move, the confidence interval, the low level and the high level of the confidence interval. And on the y-axis, we have the expected augmentation or increase of introgression in China compared to France. And in all cases, we obtain, or simulations are within the confidence interval of expected increase in China compared to France, just because branch expansion. So we could explain this higher level of introgression in China compared to France without the need of invoking selection. So the second and final case study is about European wildcats. So this is about the asymmetric introgression between different taxa, higher in the invasive and lower in the locals. And European wildcats were considered to be almost extinct or extinct in Switzerland during about 30 years. But by the end of the 60s, Switzerland changed a policy for protecting forest habitats. And by doing this, the few individuals of wildcat start to reproduce and they start a ranch expansion. So they are considering ranch expansion during the last 50 years. So in a first paper, we first demonstrate that an history of ranch expansion is the best explanation for the level of introgression or gene flow between wildcats and domestic cats. And in a second paper, we improve a density-dependent range expansion model for explaining this level of introgression. And then in a third paper, we're trying to explain or project what may happen here for the near future. So we are collaborating with colleagues at University of Zurich. So they are monitoring European wildcats during the last decade, so since 2010. So we have sticks, many sticks in the Jura Mountains, like this. So here is my colleague waiting for killing a cat and take the sample. No, it's not true. It's just helping the stick to collect the sample of wildcats. And for a reason, I don't know the reason, but wildcats really liked valerian water. So we use this valerian water to attract the cats. So in this video, you will see a feral domestic cat that escaped home and went to the forest. And so you will see in a few seconds how this cat approached the stick, and then my colleagues collect the samples for the genetic analysis. So in a few seconds, you will see wildcats, an European wildcat in the Swiss Jura. But unfortunately, this cat was more attracted by the camera than to the stick in this video, but usually it's doing the same things. And you will see that not only cats we are interested by stick in the forest, so we also have the samples of other animals too. So now, so this is the expected level of this is the observed level of introgression for different markers, autosomal markers, mitochondrial DNA, and Y chromosome. And in all cases, the level of introgression is higher in wild cats compared to domestic cats. So this is the neutral expectation for wild cats in branch expansion that I explained earlier. So then we perform simulations again. So we have on the x-axis the interbreeding success rate parameter, the frequency of admixture between both cats for different kind of markers, the prior distribution into dotted lines, and the posterior distribution into solid lines. And we test two different models with or without competition between both cats. And this budget factor says that the model without competition is the best one for explaining the level of introgression between both cats. So we use this model for projecting what may happen in the near future. So on the y-axis, we have the level of introgressions. On the x-axis, we have the time in generations. And in 100 years, we expect a massive introgression from domestic cat into European wildcats, about 60% for autosomal markers, almost 80% for the mitochondrial DNA, almost 70% for the Y chromosome. But then we may say, okay, but Switzerland is doing pretty well in protecting cats, so we may expect that in the near future, the effective population size of wildcat and domestic cat will be equal. So even in that case, we expect a massive introgression in wildcats, almost 50% for the autosomal markers, almost 60% for the mitochondrial DNA, and more than 40% for the Y chromosome. Well, we could say, okay, Switzerland is doing really, really, really good. So we may expect the near future to have an effective population size of wildcat much higher than the one of domestic cat. But even in that case, we expect this massive introgression, almost 40% for the autosomal markers, 50% for the mitochondrial DNA, and 35% for the Y chromosome. And the only scenario in which we have no more introgression is when we prevent more interbreeding between both cats. But now we could be really optimistic and say, okay, competition was not important in the past, but it will be important in the near future when this population reach a given equilibrium, and this competition will favor wildcats because wildcats are more aggressive and they have a better knowledge of the forest habitat in the Jura. And even in that case, we expect this massive introgression. And the only way of avoiding this in the short term is having at least an equal effective population size between both cats. And for avoiding this in the long term, the effective population size of wildcat should be much higher than the one of domestic cats. And again, the only scenario in which there is no more introgression is when we stop interbreeding. So preventing further hybridization could be a priority. The level of massive introgression I'm showing here actually already happened already happened. Already happened in Scotland. The integration in Scotland followed this massive integration during the last century, and was a very in a process that happened in less than 100 years, like this. And today, the question is what we do with the hybrids in Scotland, because there is no more hybrid, they are all introgressed. So and for avoiding this in Switzerland, probably also in France and Germany, we should do something. And because in our simulations, the mitochondrial DNA is the most affected marker, any conservation management program could target the females living in farms or in areas close to a forest. So, as a summary here, the observed level of introgression is explained as a ranch expansion of wildcat in the Swiss Jura. What is interesting in this case study is that invasive species in our theoretical expectation is not necessarily an exotic species. It could be a native organism threatened for many reasons, but which is in range expansion because the implementation of a conservation program, but this conservation program could result in the unexpected output of genomic swamping, so genomic replacement of the local population, the native population, because we don't take measures concerning the hybridization with an invasive, exotic, domestic organisms as domestic wildcats. So now I will say something about the implication of all of this in conservation genetics. So 20 years ago, there is a very nice paper published by Allendorf in which he provides 3 categories of hybridization to provide priorities in conservation biology. So the first one is unfertile hybrid. There is with no introgression because hybrids are unfertile. And the second one in which introgression is widespread into population, or the third population in could be the case of wildcats today in Switzerland, or fertile hybrid with complete introgression, which could be the case of wildcats in Scotland. But I propose new categories, and the new categories, the first one is my hybridization type 1, which is the same as Allendorf with unfertile hybrids. And the third type is hybridization with introgression, with gene flow, which is like the case of wildcats. What I propose as second category, sorry, I will say something about second category now soon. But in my previous papers, with other case studies, I demonstrate that the hybridization type 1 could represent a demographic threat, and hybridization type 3 could represent a genetic and a demographic threat for many species. But there is a second type of hybridization in which hybrids are fertile, but there is no introgression because the offspring produce clonal gametes of one of the parents or the 2 parents. So in this case, there is no introgression, but extinction could happen very fast because you could have a replacement, a direct replacement from one of the 2 parental species. And in another paper, we do a review of these case studies, and we observe 80 taxes having this type of hybridization in animals, most of them in amphibians, insects, and fishes, and there's much more in plants, but they are not a botanist, or I don't know, but there are much more in plants. So as a summary, there is these 3 types of hybridization. Hybridization type 1 is not a priority in conservation biology when there is no observed demographic decline, but it's a priority when you have a demographic decline. Hybridization type 2 is always a priority because extinction could happen very quickly. And hybridization type 3, it could be a priority, a management priority, if you have an observed ecological effect. So for instance, if you have a domestic organisms that change the behavior, the habitat use of a local population that you would like to preserve the ecological function of that species. But it's not a priority for conservation biology, or it could even be a tool for conservation biology if the 2 species or populations are closely related. So if they are closely related, phylogenetically closely related, and they have similar ecologies, hybridization, even if it's motivated by humans, could be a tool to increase genetic diversity and the opportunity of adaptation to change, to changing environmental conditions. So finally, I would like to say that my research is at the crossroad between population ecology and population genomics, and my aim is to help one day to fill a gap between these two fields of research. The eco-evolutionary models we are working are useful in evolutions to reconstruct the history of the evolutionary history of any taxa, but it's also important in conservation biology because provide an opportunity to project what may happen in the future with different conservation strategies. And this could be applied to other research fields. So for instance, in agriculture, if you would like to analyze the spread of a genetic variant, or in biomedicine for antibiotic resistance. So finally, I would like to thank the different people related to my different projects, Matias Cura and Juan Montoya Burgos from the University of Geneva, Logan Escoffier from the University of Bern, Tim Coulson, Sonia Clegg, and David McDonald from the University of Oxford, the Swiss National Science Foundation that support me in different projects, the University of Fribourg, Oxford, and the anthropology units of University of Geneva, where I did my PhD. Thank you very much.
Thanks for the very pedagogic talk. Very nice. Is now time for questions, discussion?
So how flexible are these models that we use to do? So I use them for, let's say, evolution of dynamics, like in cellular organisms, like no bacteria or whatever, like can you transfer it, or is this very specific for this like interspecies?
Okay. Yeah, it's not so easy with, okay, Splash, the spatial species software is for diploid organisms. And my general model of hybridization too. But the R package is also for diploid organisms. And it's very specific for but you could, it's easily to modify the R package. So in that case, we implement a fitness function. So everything is there in the fitness function. So in the paper, I provide a few examples of fitness function, but I have to develop this. But I provide like 5 fitness functions for different cases. But the idea is that people little by little start to think about different life histories, different interactions between different kind of organisms, and eventually I think it's quite flexible. But you need to have a knowledge for the fitness function. You could implement this in R, but if you would go, if you would like to do it like this in more detail, probably you can change this, the base of this in C++, but it's not so hard to change. But what I would like to say that finally is flexible, the R package, but we will see which case study if people are interested in the package, start to develop.
So population species is like, it's like nice to think about it, but in a way it's like also impossible to keep the species that you want to preserve separate from the other species. So I mean, it's a bit of a philosophical question, right? But like, it's impossible to prevent the mixing, right?
Yeah.
Probably physically separate them.
Yeah, it's a very nice question because actually a main problem of conservation, but the old version of conservation biology is like preserving a system as a museum. And systems are not a museum. Systems are dynamics. So you have eco-evolutionary things happening all the time. So what I try to highlight with these approaches is we cannot remain in a status quo, but what we could do is try to have less influence in natural systems, and for maintaining ecological. Yeah, but it's also philosophical. It's also philosophical. For me, it's important to maintain the systems working with the evolutionary potentials and everything. So, for instance, the wildcat case study. The wildcat case study, they are declining, and you have the hybridization. If the hybridization is not changing much more in the behavior of cats, this could be a positive thing because you are increasing genetic diversity. And for instance, in Scotland, the wildcat disappeared, but you have the hybrids. So the legislation in Scotland, you have to kill the hybrids and you have to kill the exotic organisms, in Switzerland too. But if you don't have any wildcats because they are extinct, you only have the hybrids, someone has to provide the role of a predator in this system. So the hybrid could do it. But the problem is with the wildcat is not so straightforward. It's not like that, because the wildcat, the domestic cat are domesticated. So they change the behavior of the wildcat. So the wildcats, they use forest habitat and they don't approach the humans, they remain there. But the domestic cat, they can go everywhere and they predate everything. So this is the main problem. So, well, it's a philosophical question, but yeah, personally, I think it's important to maintain the dynamics working. And this kind of model will help you to project what may happen.
Do you consider involving more or
Okay. Splash is restricted to 2 different species or 2 different types, but we are working now in a new version for 3. So this is nice. The general model of hybridization that I did during my PhD is also for 2, but they are packaged. You could simulate many, say, how all the species and populations you want. You could have many if you.
To continue on this question, I'm thinking that you will have eventually kind of 2 species with a hybrid. In Splash, for instance, you count on every person back to 1.
Yeah. And this actually happens. There is a very nice case of rabbits, kind of rabbit in Europe, in which you have introgression in the current. So today you have introgression from a population that never meet the other populations. So it's like they hybridize with another species. Then they current these genes, and like in the middle you have a third one, or second or a third one. That was a bridge of gene flow between the 2. And in the case of modern humans, we would like to apply this with Neanderthals and Denisovans. So Neanderthals, Denisovans, and modern humans. But this complex is a complex thing. So with the model, we would like to simplify as much as possible, but sometimes it's important to have these interactions. So for instance, if we would like to know what happens with human ancestry we have today during the last 1,000 of years, it could be nice to have the interaction between Neanderthals and Denisovans with the modern humans.
Okay, so I think we can continue the discussion with some pizza.
Yeah, thank you.
Thank you for coming. Thank you.
Thank you very much.